Okay, time to start. On behalf of the Institute of Agriculture of the University of Western Australia, we warmly welcome you to come to this seminar of the 2023 Lefroy Fellow, Dr. Kelsey Pool. We acknowledge that the University of Western Australia is situated on Noongar land, that the Noongar people remain the cultural and spiritual to practice the values and languages. We are here with Kelsey because she is the Lefroy Fellow. She was awarded the fellowship, I think 2 years ago, Kelsey, and is continuing her research for another 1.5 years. I am, and we all are, I think, very interested to hear what she has found. She will explore her latest research and has established a core research program devoted to improving sheep production. E.H.B Lefroy was trying to do nearly over 100 years ago now. And that's expanded to a livestock reproduction business and large-scale model for human infertility. We'll hear about how diet can change the way sperm functions, how in-utero events can alter the physiology and even behaviour. All pretty impactful things of future generations and how we can use markets of fertility as a bioassay for biological de-fleecing. But prior to hearing what Kelsey's research, professional research, is about, I'd just like to cover a little bit about her personal interests. Kelsey grew up in Sydney, on the outskirts of Sydney, and her education was very much part of in the University of Sydney, where she did animal and veterinary bioscience, which she found an absolutely awesome degree at that time. She found it incredibly hands-on, and she actually credits doing that degree to bringing her into contact with agriculture, and she has a natural practical disposition. She is married to Mark. I think Mark is here. Hi Mark, welcome. Who is a chef of some renown and held in a very highly professional state in Western Australia. He is a very big support for Kelsey and the time she is giving to her profession. Outside of her professional work, Kelsey also has other creative interests. She is an artist of some renown. Now this here is a drawing of Mark. And only today, the Archibald Prize winner was announced, Julia Gutman, but I think she would have given her a run for her money. And when I was at university 50 or so years ago, you'd all know probably of Henry Schaffer. And I was fortunate enough to go to an address that Dr. Schaffer was giving in Winthrop Hall about education. And what he said was, the end point of education is creativity. And I think it's so exciting to know that background of you, Kelsey, as well as that of your professional background. One of Kelsey's other keen interests is that of underwater diving. She loves the reefs, finds the ocean and the seashore, if you like, stimulating and refreshing. So she's here probably for quite a long time, about which we're very fortunate. Her practical scientific aim is to both contribute to a better mechanistic understanding of reproductive biology, whilst providing tangible outcomes to the Australian livestock industries. Her research focuses primarily on understanding how endocrine disruptors and environmental factors impact fertility through both multi-generational Drosophila melanogaster models and sheep production systems. She completed her PhD at Sydney, but also went to INRA, I-N-R-A, which most of you will know about, Institut National de la Recherche Agronomique at Neuilly, near Tours in France. It's a bit like a central bank, with a central currency of curiosity floating around the whole place. It must have been a very exciting time, and I'm pleased to say that our Dr. Graham Martin, Emeritus Professor Graham Martin, also was there for some time, so I can imagine what a stimulating environment that would have been. Her PhD research into how the neurohormone melatonin regulates reproductive seasonality resulted in the registration of a commercial Australian product to improve ovine reproductive efficiency. She strongly advocates for better integration of applied and fundamental research in the field of agricultural science, with particular emphasis on involving producers in research and the benefits of livestock as models for mammalian fertility. Ladies and gentlemen, my great pleasure is to welcome Dr. Kelsey Pool.
All right, thank you, Phil, for a really lovely introduction. And from me personally, welcome to everybody here tonight. I am so thrilled to see not only my UWA colleagues here, but we've got some from CSIRO, we've got DPIRD here, some from Murdoch University, University of Sydney, which I'm still a little bit shocked about. That was a surprise one. Producers that I have worked with across Western Australia, and some non-academic friends here as well. So look, thank you so much for turning up tonight. It really does mean the world to me that you're taking your Friday night to listen to me talk about reproduction. While there are a lot of you that I do know here tonight, there are quite a few more that I don't. So a little tiny bit more of an introduction on me and why we're talking about infertility tonight. You've probably gotten the gist that I am the 2020 to 2023 Lefroy Research Fellow. And it's a position that I actually took on before I'd even finished my PhD at the University of Sydney. I am a reproductive biologist by training, with a fairly heavy focus on male fertility. Since arriving here at UWA, I have been nicknamed by mentor and boss Dominic Blash as Dr. Sperm. It's a title I wear with pride, and you'll see why I've earned it, hopefully over these next couple of slides. What's been interesting about my career in reproductive biology is that, unlike a lot of my fellow reproductive biologists that are really focused on the fundamentals of human medicine, my training in this field has been almost entirely in Australian agriculture. And this is a field I absolutely love to work in. I think it's fantastic that what we're doing at the university level and at the lab level, we're able to actually translate to on-farm outcomes, and we can see the impacts of this research within a couple of years. Certainly not always an easy space to work in. Sometimes what I do is too livestocky or too applied for the more fundamental reproductive biologists. And on the flip side of that, sometimes what I'm doing is just too molecular to be of direct interest to industry. However, I think that these two parties are actually far more closely intertwined than what they both realise. And in fact, one of the most interesting things that I have seen through my career so far is that these issues that I'm investigating in emerging issues in livestock reproductive health actually parallel, or sometimes even foreshadow, what we see in human fertility. And it begs this question: are we actually overlooking a really valuable aspect of livestock research? Aside from the fact that these systems are so valuable to study in and of themselves, could we also be using these populations to model human infertility? Now, with 8 billion people on this planet, maybe you're wondering, is there even an issue with human fertility? I think as a species, we're fairly fascinated by our own ability to reproduce. Trust me, there are whole legions of researchers just looking into the fundamentals of this. Which is why it's interesting that when we realistically think about the future, we think about what our climate is going to be like, how we're going to change, how we'll evolve and adapt. We think about what technology we'll be using, what our cities will look like. What we fail to capture realistically in this image of the future is that at the rate we are travelling, very few of us are going to be able to naturally reproduce. I'm certain that every single one of you in this room is aware of the fact that we've just lived through a global pandemic. Every single one of you will know something about the events of COVID-19. Maybe you know how the virus causes disease. Maybe you've had coronavirus and you're aware of that fact. Maybe you've been vaccinated for it. What most people don't realise is we've actually been living through another pathological event of pandemic proportions over the past few decades. Since the 1950s, we've been seeing a very steady increase in mammalian infertility. Now, infertility is incredibly common. This pandemic, this silent pandemic, is one that we're not talking about. As it stands, 1 in 6 couples are currently considered to have some form of infertility, and about 1/3 of these cases are attributed to the male side of things. Now, we've been seeing an annual 1% increase to reach this point. So at the rate we are travelling, we're not talking about a gentle population curb, which arguably I think most of us would think is a good thing. We're talking about a detrimental decline in our ability to reproduce, a heavy reliance on reproductive technologies, and an overall decrease in our reproductive health, which is of course linked to our systemic health. We are not just here to breed. So how on earth did we get to this point? I think the answer to that, at least in part, is the fact that our modern world actually represents an incredibly hostile environment to our reproductive health. On a daily basis, we are exposed to these compounds called endocrine disrupting chemicals. And these compounds interfere with our hormone signaling. They change the way our bodies function at a systemic level. These compounds are ubiquitous in daily life. They are in the air we're breathing, they're in our waterways, our diets, our personal care products, our cleaning products, and in the plastics that we're making our children's toys with. And we've got pretty good evidence that they're challenging our ability to reproduce. Now, I'm particularly interested in dietary endocrine disruptors, and that's because these represent a problem not only for human reproductive health, but also for livestock reproduction and even reproduction in our wildlife species. In the average pantry, you'll find everything from dioxins to phthalates to phytoestrogens. These endocrine disruptors contributed to our diets from everything from the packaging around our food, the residues on our fruit and veg, and even plant estrogens or phytoestrogens in the fruit and veg themselves. And it's these phytoestrogens that we're really going to be honing in on today, because again, they're a very common example of how an endocrine disruptor can change not only human reproductive health, but also the ability of our livestock to reproduce. There's another really scary part to this story. We're starting to get pretty good evidence that these compounds can damage the reproductive potential of that exposed individual. But even when we're reducing this exposure, we're still seeing this persistent increase in infertility. What if this subfertility or this infertility can actually transcend that exposed generation? Now, we don't know how this changes, if it's a maternal line of exposure or if it's a paternal one. What we do know is that to collect this data in humans or even in livestock, we're looking at anything from several years to several decades. By the time we start to understand the mechanisms underlying this event, we are going to be too late to find a solution. Now, fortunately, when I arrived at UWA, I actually had access to a really powerful modelling tool for multi-generational fertility. And that's a little species called Drosophila melanogaster, also known as the fruit fly. Now, I've been really sold generally on the fruit fly as a model. I actually use it to direct my livestock research now. With a rapid generational turnover, a relatively low cost to run, and genes and biological mechanisms analogous to what we see in mammals, these are a much lower cost and lower time intensive way to start to test these experimental ideas before we take them to the field. So in this case, I wanted to investigate what would happen if we included a really common phytoestrogen, a dietary endocrine disruptor, into the diets of these fruit flies to see what was happening to their reproduction. Now, we're not talking about crazy levels here. This is similar to what would be found in a Western diet or perhaps in the diet of livestock grazing an estrogenic pasture. So we exposed these fruit flies in what we call the P1 or the parent generation, and then bred these to form the F1 or children generation, and then F2 or the grandchildren of these exposed individuals. What's important to note here is that beyond this parent generation, these subsequent animals have never encountered this estrogenic compound in their lifetime. And then we went on and measured their fertility. Now you can see we've got distinct lineages here of either maternal or paternal exposure, as well as what we've dubbed the double exposure lineage. In this first generation, as you might expect, we see fertility reduced across the board in these exposed individuals. And when we start to look into genes controlling or regulating important reproductive processes, things like spermatogenesis and embryo formation, we find that these are all downregulated in these exposed individuals. Now, this may go some of the way to explaining the infertility in these exposed individuals, but this infertility actually persists into that next generation. These genetic changes do not. This is true regardless of whether you're a male or female offspring from an exposed female or male parent. The only case where this is reversed is in that double exposure lineage. Now, what we suspect is happening here is we're applying such an intensive selection pressure that these animals that are able to reproduce are in some way superior. Maybe they're able to metabolize equol a little bit better. Maybe they're simply not eating as much of that diet. We don't know. What we do know is that by the time we reach the grandchildren, fertility is again reduced across the board. There was something else really interesting going on with these fruit flies though. In a normal fruit fly mating, you should be seeing about 50-50 boys and girls. It's usually about a half-half on that sex ratio. From the first point of exposure, we start to see evidence of a female survivor bias. For every one male, we've got 2 females. Now, this actually becomes far more evident as we travel down those following generations. This is a fairly common event in nature when an environmental stressor is applied. Biologically, it makes more sense to invest in a female offspring from a genetic perspective. A dud male with poor genetics is potentially affecting several females. This did lead me to wonder, however, what was going on with these surviving males. We know their fertility is reduced, but what's actually causing this? Now, my first port of call to do this in a mammalian species would be to look at sperm function at a molecular level. The sperm cell is a really brilliant indicator of male reproductive health broadly, and it gives us quite a lot of insight into the function of that male reproductive tract. However, I quickly realized that these tools that I would normally use didn't actually exist for fruit fly sperm. And so this meant redeveloping and optimizing 6 new assays to actually measure sperm function at a molecular level in the live sperm cell in real time. The kinds of things I am interested in are the status of lipids in the sperm membrane. Are these disordered? Are they undergoing lipid peroxidation? I'm interested in the sperm DNA, that crucial genetic material for contributing to the next generation. What is the stability of this DNA? Is it prone to fragmentation under stress? Anybody who has done biology to any degree will know that mitochondria is indeed the powerhouse of the cell, and this is very true for the sperm cell. This organelle is responsible for driving that tail movement, driving that cell through the reproductive tract. In the fruit fly sperm, we can now measure electron transport across the membrane of the mitochondria. We can see specifically if this is undergoing oxidative stress. In sperm, when we see these changes concurrently, it generally suggests that this cell is under intense metabolic stress. It is undergoing apoptotic-like changes, or essentially cell death. And that's exactly what we see in these exposed males in the parent generation. Purely after consuming this equol-rich diet, they've actually got 50% less viable cells than their non-exposed counterparts. That's 50% less cells able to contribute to a successful embryo. And this actually persists in their children. These subsequent males have still got this phenotype of metabolic stress. They've still got 50 to 40% less cells alive. What's really interesting here, however, I think, is the sperm of the grandchildren. because in 5 of 6 of my assays, these cells appeared completely normal. They're around the same viability, they're not undergoing this metabolic stress. What they did have, however, was a silent but deadly defect. The DNA in the nucleus of these cells was incredibly unstable and prone to fragmentation. And it's a silent but deadly defect that could easily be missed in a clinical setting. For example, if I'm assessing a livestock semen sample for fertility, unless I am specifically asked, I don't routinely perform that DNA fragmentation assay. It goes a long way to explaining some of these cases of male infertility in otherwise completely healthy males. So the message of this study is a bit of a sobering one. We can try and protect our fertility by reducing our exposure to endocrine disrupting compounds. But our reproductive potential may have already been pre-programmed by the diets of our grandparents. Now, while the fruit fly are a brilliant model to look into things like transgenerational fertility, it's much more difficult to get an idea of the biological mechanisms at a tissue level. We want to understand what's actually driving these events. Now, arguably, we don't actually need an animal model for dietary phytoestrogens and fertility, because we've already got a prime example of that going all the way back 80 years ago. What I'm talking about is something called ovine clover disease. Now, ovine clover disease essentially describes female sheep grazing these subclover-based pastures, and these subclovers were really rich in these phytoestrogens or plant estrogens. These animals had far poorer fertility, and it's a problem that still persists today, although certainly not as rampant as it was about 80 years ago. This is actually the project that I was contracted to come and work on here in Western Australia. I was meant to be coming here to look in the mechanisms driving this infertility in female sheep. However, given that my background is in male reproduction, I had to ask the question, well, has anybody actually looked into the rams? Has anyone checked the boys? And it turned out for the last 80 years that no one actually had. Ovine clover disease was considered to be a purely female-oriented issue. Now, given what we know about estrogen and male reproduction generally, this hormone has a crucial role. This never rang true to me. So I decided to run a little bit of a mythbuster study and just see if this was actually the case. Are our rams in fact safe on these estrogenic pastures? Because from work 80 years ago, this was actually the recommendations around these pasture use. So we essentially ran a study at UWA Ridgefield Farm. We were comparing the reproductive capacity of these rams either on a control pasture or one with these estrogenic clovers. We were measuring just about everything we could estimate to look at their fertility and reproductive potential, but to also understand the mechanisms underlying these events. We followed up in the following breeding season as well, because I think a really crucial part of ovine clover disease is knowing whether that infertility is transient or permanent. Now, just like in our fruit flies, it turns out that these phytoestrogens certainly have the ability to alter ram reproduction. But for the first few weeks of the trial, we actually didn't see any difference between our treatment groups. It wasn't until around 5 weeks of these animals grazing these estrogenic pastures that we started to see something a bit odd in the sperm of these rams. Now, in a normal ram sperm sample, we should be looking at less than 10% abnormal cells. So in this example, we've got about 5 abnormal cells in a field of 200. What we were seeing in these rams grazing these estrogenic clovers was about a 7-fold increase in abnormal sperm cells. And interestingly, it was only one very specific abnormality. If you can all imagine that classic tadpole shape that we associate with the sperm, what was happening to these cells was, rather than that nice straight tail, this was coming off the sperm head at a right angle. These cells were still motile, but essentially they were swimming in circles, so not particularly useful for swimming through the reproductive tract. Just like in our fruit flies, we also found that the sperm DNA was incredibly unstable and prone to fragmentation. But we really wanted to understand what was going on, what's actually driving this event. So we took a proteomic approach and looked into proteins in the seminal plasma. And this is a really great way to indicate what mechanisms are actually driving this event. We got back a whole list of proteins that were differentially expressed between these two groups of rams. Things with organelle construction and membranes, things about immunity, apoptotic DNA fragmentation. But what we need to remember here is we're not just talking about a list of differentially expressed proteins. These proteins represent a dynamic, constantly interacting network. They perform in concert to cause specific biological functions. And when we started to apply a bioinformatics approach to actually look at them in a more dynamic network and a more holistic way, we actually start to see something really interesting. These proteins have one biological function, and that's purely concentrated into spermatid formation, or the final stage of spermatogenesis. Essentially, this stage describes the formation of that tadpole shape. And these proteins that were misregulated in these rams were responsible for things like mid-piece assembly formation, the attachment of that tail to the sperm head. They were responsible for the packaging of that sperm DNA and the cytoplasm removal to ensure that correct shape. What we can say with reasonable certainty is happening here is that we know that estrogen receptors are greatly increased during this stage of spermatogenesis. In the presence of these plant estrogens or phytoestrogens, we actually see a competitive binding to these receptors to misregulate these proteins and cause this bent sperm phenotype. So what does this mean in an industry sense? Well, if you are joining on pastures where there's no active growth, you should be still relatively safe in terms of fertility. We found that these effects were transient once these rams were removed from these estrogenic clovers. And in WA, we don't generally have a lot of active green growth around joining. However, if you are using your rams out of season, so people like stud breeders, cryopreservation of semen, use in controlled breeding programs, then we certainly want to avoid grazing our rams on these pastures. In terms of ovine clover disease, for the first time in 80 years, we've actually contributed another facet to this story. We've shown that this is not, in fact, a purely female-oriented problem and is, in fact, contributed to by both the sexes. As a model for human fertility, this study actually represents the world's first longitudinal study of how dietary phytoestrogens contribute to male infertility and provides a mechanism to describe this. Now, fortunately, these studies into livestock reproduction are not always about just finding a mechanism as to why reproduction is reduced. Sometimes they also become really awesome opportunities to actually look into solutions. And this is exactly the opportunity that I had in the second year of my postdoc when I was asked to investigate solutions to improve twin lamb survival. Now, for those of you outside of the field of sheep reproduction, and there are a few of you here tonight, twin lambs probably seem like a bit of a production win. You're essentially getting two animals from one. Obviously, you need to put in a bit more resources to this ewe, but you've essentially got a fairly clear production gain. However, just like with human twin pregnancies, these extra lambs come with a lot of extra risk. These animals are more likely to experience hypoxia, this reduction of oxygen flow to the brain and other organs. They're more likely to undergo these prolonged, traumatic and potentially injurious births. And we actually see things like edema around the head and rib cage and even hemorrhaging of the internal organs. They're born at lower birth weights with less energy reserves, less brown fat, and because they're born into these fairly harsh winter conditions, they are far more prone to death by exposure. Now, all of these factors together actually contribute to an average of a 30% mortality rate across the average sheep production system. So suddenly that production advantage has disappeared, and we've got a fairly horrendous animal welfare issue on our hands as well. What's quite interesting about this, however, is that these animals that do survive all of this are then potentially faced with a lifelong impairment of cognitive function. We'll talk a little bit more later about why cognitive function is important in sheep, But what I want to draw your attention to here is that this is actually something that also happens in humans. And it's something that's incredibly difficult to study. I will note that I have to tread quite carefully here. My husband, who we've already drawn attention to tonight, is also a twin. I just reassure him on a daily basis he's not the hypoxic twin, so I should probably be apologising to my brother-in-law. So what we wanted to do was to find a preventative solution to this. Could we actually protect these animals by treating them before they're even born? Essentially, by treating them in utero or treating the mother. And our best candidate to do this was a neurohormone called melatonin. Now, melatonin's already therapeutically available in humans, and it's also commercially registered for use in sheep. In sheep, it's generally in the form of a slow-release implant, and it's used to improve reproductive performance outside of the breeding season. In this case, we were looking at treating these twin-bearing ewes, for example, during early gestation. We're using these antioxidant properties of melatonin, these vascular growth promoting properties, to try and support all the development that's occurring here. We looked at using this during late gestation, aiming specifically for that birthing process. Could we protect against hypoxia, as melatonin's a really effective neuroprotectant? Finally, we also treated these animals during essentially what we called whole pregnancy, so because of the slow release nature of these implants, from early gestation onwards. Now, fairly classic of just about every study I seem to run, we've measured everything we could get our hands on to try and come up with some kind of strategy to protect these twin lambs. Now, one of the first things we can actually see is that just by treating these animals in utero, we're actually manipulating and changing the vascular communication between mother and fetus. What we're looking at here is the umbilical cord insertion into the neonatal liver. And we actually find that in these animals treated with melatonin in either early or late gestation, that this vascularity is actually increased. We do see this effect reversed to some degree in these whole gestation exposed animals. That's a fairly typical property of melatonin, where it has a dual role. At a certain threshold, its role in vascular communication reverses. Now, this finding goes some way, but not all the way, to start explaining the physiological differences we then started to see when these animals were born. What I can tell you about these melatonin-exposed lambs, particularly those exposed during late gestation, is that they are built different. In a production perspective, by weaning, we start to see that they're on average 1 to 3 kilograms heavier than their non-exposed counterparts. This may seem like a fairly subtle change, but if we then combine that with the 6 to 8% increase in survival that we were seeing, this is actually a significant difference. These animals are born with more brown fat around their heart and kidney, that important energy reserve required for getting through those first few days of life. When we look at their immunity, specifically immunoglobulin G, a really good indicator of an animal's ability to fight infection, we find that this is vastly increased in these late gestation exposed animals. Their ability, potentially, to fight off an infection is far increased. In collaboration with Curtin University, so a special thank you here to Dr. Mark Hackett and Dr. Rhiannon Bosley, we actually formed biochemical maps of neonatal tissues to try and understand what was going on at a macromolecule level. What we started to see indicated that these animals also had a more rapid metabolic turnover. They've got this improved defense system against lipid peroxidation. Essentially, these animals were born with a physiological advantage. What was really interesting about them, however, was actually their brains. Despite being similar dimensions for body weight, we were actually finding that their brains were heavier. And while we're still in the process of measuring it, there's also a potential difference in the populations of neurons in regions of learning and memory. Which brings us to cognitive function. Now, this is something I was really quite skeptical about in animal studies. I think it's something that's really hard to objectively capture. However, I have been converted. We were looking into tests for things like learning and memory. How well can these animals navigate a novel environment? How do these animals respond under stress? Do they vocalize differently? Do they communicate differently? How do they interact with that environment? Well, in collaboration with some of our colleagues in China, we've actually been developing a program to more objectively quantify this. We can now use artificial intelligence to more accurately record how these animals are behaving in an environment and give these more objective scores for assessing behaviour. What we started to see in these animals, again, anything that was exposed in late gestation, is that they actually appear to have better cognitive function. These animals show a far greater learning potential. They're able to actually successfully navigate these environments much more rapidly than their non-exposed counterparts. They communicate differently. They are doing less stressed communication, more likely to communicate with animals in the testing arena. And overall, in response to this novel and potentially stressful environment, they actually have a lowered stress response. And we know this both from their vocalizations and from plasma cortisol. Now, I'm certain you can see why these attributes would be important in human children. Everyone wants to think their child is a superior learner. But why is this important in sheep? Well, the sheep that's got a greater learning capacity is more likely to be able to navigate the yard successfully rather than turning around the wrong way. Animals that can vocalize and communicate with other members of the flock are going to be more likely to be able to identify their mother in a flock of several hundred similar animals. They're more likely to alert to a predator if one is near. We know from numerous studies completely outside of my field that a lower stress response is just generally beneficial to animal welfare and production. These animals tend to produce better, they are more successful at reproduction, and it even improves meat quality. So what we can take from this study is that by applying these slow-release melatonin implants during late gestation, we can actually produce offspring that have a physiological advantage. These animals have got greater energy reserves, a better immune response, their live weight is increased, and they actually appear to have superior cognitive function. Now, I suspect melatonin, like just about every therapeutic alone, is not going to be a be-all or end-all solution. What we need to be looking at is combining this with more environmental management. How can we manage this lambing paddock environment? How can we manage maternal nutrition? And I think these factors together will really help us boost solutions like this. What this study really drove home for me, however, was that this period of life, this late gestation to early neonatal period, is actually crucial in driving how we behave, or in the case of animals, how they produce as an adult. And as a reproductive biologist, this is actually an area that's really inaccessible to us to measure. If we wanted to look into things like fetal physiology, we could put these animals in an animal house and we could apply all kinds of relatively invasive measures. We might get a lot of data that way, but we're losing that commercial relevance. We can't say what's happening to this animal in a field environment. Imagine if we could objectively measure fetal physiology on farm. We could get information into how handling of these late gestation pregnant ewes affects the ability of their offspring to produce. We could know about the effects of things like endocrine disruptors during late gestation, effects of melatonin, how climate change is impacting these animals. Imagine if we could do that. Well, that's exactly what we've actually been developing across a collaboration with Murdoch University and University of Western Australia. What we have designed is a completely ultrasound-guided laparoscopic keyhole procedure to place a microbiosensor into the ovine fetus. These biosensors log things like fetal temperature, activity, and heart rate. So from this information, we can understand more about circadian rhythms and fetal stress. This procedure is designed to be performed on farm. After a relatively rapid recovery time, this ewe would be able to go back into that commercial production system, give birth naturally, and continue her normal commercial life. Similarly, we wouldn't be collecting that logger from that lamb until it actually went to the abattoir. So currently, what this looks like, just to kind of humble all this down, this video here is actually showing us in a cadaver model, probing the fetal abdomen to find the safest place to put that logger. We've all had a pretty big learning curve in our ultrasound skills, and so I always have to put this picture up here to kind of show how far this project has come. I think our expressions here perfectly capture how difficult this project was at the start. It's been an enormous learning curve. I do have to say a really special thank you here to Dr. Serena Hancock, who has actually made it tonight, which is awesome. She has been my sonographer for this project since day one, has turned up to every single farm training event. And also to Dr. Scott White from King Edward Memorial Hospital. So Scott is actually a fetal laparoscopic surgeon that's really been helping us refine this procedure to be as safe and rapid as possible. In more exciting news, we've actually gotten an invitation from the University of Cambridge in the UK for me to travel over there and actually teach them this fetal surgery. So I think that's pretty amazing, and I think the implications for animal welfare in these kind of studies is really important. So I'm really glad that this is being picked up on an international level. So hopefully what I've sold you on today is that these livestock systems actually provide not only valuable research opportunities in and of themselves, but also quite interesting models for mammalian fertility broadly. However, I think what we lack here is a really good integration between industry and academia. How do researchers actually pick up on these issues in a timely manner so we can actually use them as sentinels for human reproductive health? Well, one way I've actually been able to do that is through starting a commercial business here at the University of Western Australia. In collaboration with colleagues at the University of Sydney and the University of Queensland, we've actually formed Australia's first internally standardised university collaborative livestock fertility testing business. And outside of being a really interesting enterprise in and of itself, it's actually provided a really valuable opportunity to pick up on reproductive issues before they become widespread. So I guess what I wanted to draw your attention to here is that these commercial ventures actually hold incredible value to our research. These opportunities are not just businesses, they are a better way to actually integrate these brilliant producers that we're working with into our research, because that's really where a lot of our ideas are stemming from. Now, it's this interest in the commercial side of things that is really my motivation for our upcoming project. And it's actually one in biological de-fleecing. Now, for those of you that do know what biological de-fleecing is, you might be thinking that this is a little bit out of field for a reproductive biologist, but bear with me, because I promise, as usual, reproduction is incredibly important. Now, we'll start back from the start. For those of you that don't know, Australia is actually facing a shortage of skilled shearers. And unfortunately, the sheep production calendar is incredibly reliant on this event occurring at the correct time. The process of shearing itself really hasn't changed for the last few decades. It still involves a fair amount of manhandling of the sheep and manual use of clippers to actually remove that wool. Several years ago, the first sort of real alternative to shearing actually came onto the market, and it was through a product called BioClip. Now, BioClip was an absolutely brilliant concept, but it fell short in adoption potential because of some fairly severe side effects. Now, the basis of BioClip really focused on this protein called epidermal growth factor, which regulates wool growth. By injecting these sheep with a large amount of epidermal growth factor, we actually cause a tapering of the wool follicle, and this allows the fleece to be gently pulled off. As I said, an absolutely brilliant concept, and something that could really be done at that farm level without that reliance on external contractors. However, it turns out that epidermal growth factor is actually important for things outside of just growing wool. These receptors are found throughout the body at a systemic level. And when you've got receptors at a systemic level with a large amount of that protein, you're going to start to get side effects. These animals were losing weight, they were scouring, and we were seeing fairly severe effects on their reproductive capacity. Now, it's these latter side effects, this effect on reproductive capacity, that actually provides a really brilliant opportunity to start to redevelop a new product for biological de-fleecing. It turns out that many of the ligands that actually cause that follicle growth are also present on sperm. And we can actually use sperm as a really brilliant bioassay, both at the lab level and in the whole animal, as a bioassay to detect any unwanted side effects. So in combination with design of a targeted delivery system, we started to search for the perfect ligand, or essentially the perfect new protein that was more directed towards the wool follicle. We again took a bioinformatics approach to look into proteins that could interact with that EGF receptor. And when we start to cluster these into just proteins that specifically interact in the epidermis, we get a relatively small number of hits. Now, what's interesting about all of these proteins is it turns out they've got a single conserved functional domain. So we've now got several quite promising candidates, as well as a functional domain that can potentially be isolated and combined with a targeted delivery system to start to work towards a new method and a safer method of biological de-fleecing. Now, this is all sounding very promising, but we're certainly not at the level where we can just go and inject these compounds into sheep. We're simply not at that stage yet. What we can use, it turns out, is another fruit fly model. It turns out that these flies also have the same epidermal growth factor pathway responsible for what they call bract and bristle, or essentially hair growth on the fly's legs. I can already tell, having talked to the students involved, that this is forever going to be known as the hairy leg model. That's already what it's referred to. But essentially, we can still search for this tapering effect in the fly hairs. So we've currently got several fly models underway to start testing these ligands. I've got a really brilliant student assigned to this project. So I guess stay tuned in this space, not only for the sheep work, but for more photos like these. So before I start to wrap up tonight, I guess what I want to draw your attention to is the value of fellowships like the Lefroy Bequest. The Lefroy Fellowship, really at its core, was just designed to support a single postdoctoral fellow in researching the care and breeding of merino sheep, and for the last just under 3 years, that's been myself. However, I've found that these funds have gone far beyond supporting just me. In fact, I think they've supported my entire team and several core programs of work. Since starting my postdoc here less than 3 years ago, we've been able to produce 5 research publications and present this research at actually more than 10 conferences. This number doesn't include all of the industry forums and open days and workshops we've been able to speak at. It's supported not only my attendance at this event, but also the attendance of some of my students as well. And as an early career researcher, I can't emphasize how important that exposure is for our other early career researchers. In having these opportunities, our students are able to actually talk about their research. They're having this funding support their research, and it's an absolutely crucial exposure to both industry and academia as they start to form that career pathway. We've been able to get a couple of extra grants along the way, a pretty big one in sheep reproduction and climate change, and also a smaller one that's meant a colleague of mine could come and do some work at UWA, again modelling climate change, but in this case also combined with endocrine disrupting compounds. So a bit of a trending theme here. This funding has supported the running costs of 2 labs, not only my own fertility lab, but also the Drosophila lab as well, and those resources are now used by more than 20 staff and students. Alongside supporting my research directly, they've also supported the research of my students, and I've had the absolute pleasure of mentoring and supervising several honours students, masters, and 2 really brilliant PhDs. I've had so many opportunities in what was a research position to teach, everything from undergraduate lecturing and workshops through to, as I've just mentioned, that postgraduate supervision. It's meant, as you've already heard, that alongside my research career, I've also been able to start up a commercial business. And as I've said, I think this is something that's been really valuable in driving the direction of my research. Also did a bit of co-hosting on a podcast for a while there, a really brilliant opportunity to start to expose myself and expose other people to reproductive biology research in species outside of humans. I have had the opportunity to collaborate with, I think, just about every university in this state. I think the only one missing is ECU, so I should probably contact them. But But it's been so brilliant. No, on a routine weekly basis, you know, I'm working in labs at Murdoch, I'm working at different farms, I work at Curtin University. I've got colleagues, wonderful colleagues from all of these places who have just taught me so much throughout this postdoc journey so far. It's also meant on my side that I've actually been able to bring over a few of my existing collaborations from the University of Sydney to over here. People like Seva Animal Health, for example, who have supported my melatonin work since 2017, have continued to support that work even after my move to WA, and they now support some of my students' work as well. Finally, it's meant that I've been able to actually sit on numerous academic boards for some fairly high-profile reproductive societies, as well as some international journals. And when I joined these council boards, they kind of asked, well, what do you want your role here to be? And so for the last 3 years, what I've been really trying to do is to advocate for the value of livestock research in the broader reproductive biology field. I am so tired of seeing these brilliant agricultural scientists and animal science have their work overlooked because they're working in a species that aren't humans. And since sitting on these boards, I have started to see changes. More and more sheep and livestock researchers are getting invitations to speak at these big events. More of their students are able to attend, and more of their students are actually getting presentations. Now, these might seem like really small and insignificant changes, but to people like me, having your research validated in that broader field and showing that it does contribute, regardless of the species, is really important. I think it's really important for our upcoming researchers as well. So that really brings me to my thank yous for tonight. I just work with so many brilliant people, and so many people have contributed to this research over the past few years. I need to give a special thank you tonight to Dr. Tim Watts for hosting quite a lot of my agricultural work, and of course to Dominique Blash, who has been an absolutely brilliant mentor, helping me navigate the reproductive world. He's become a pretty good friend as well. He might not admit it, but he is. All of my research team will know that I can't go through a lecture without having a photo collage slide, and I never think that a list of people does it justice anyway. So here you all are. You know, thank you so much to every single one of you. When I arrived in Western Australia, you know, it was peak COVID, the borders were closed, and it turned out that I wouldn't see my family and friends for about 2 years. It had the potential to be an incredibly isolating situation, but I have found it's been the exact opposite. Since arriving at UWA, I have just formed this incredible research family. You know, they support me in the field, they help me in the lab. We laugh a lot, there's probably been a few tears in there as well. You know, you are all so brilliant. I hope I have taught you even a fraction of what you have taught me, because it's been an absolute pleasure to work with every single one of you. Now, my final thank you tonight, of course, is to the Lefroy family and the Lefroy Bequest Committee. Because without this opportunity, you know, none of this, from the science through to this research community, would have been possible. So thank you, because what an absolutely brilliant way to kickstart off a career. Thank you. We can leave it on this slide, I think, for questions.
Ladies and gentlemen, what an exciting presentation with all the possibilities that might flow out. So there's time for questions if anyone would wish to ask a question.
There we go.
Firstly, Kelsey, that's fantastic. I heard you speak down at the field day when we had in the farm, and yours was riveting. The only thing was that Dr. Ding came along later and captured my imagination.
Dr. Ding is always upstaging me like that.
It was the fact that he was talking about temperamental research. This is something I've been fascinated by for 50 plus years. And I'm just a bit interested to work out, you know, whether you see, temperamental selection, you know, is there a cushioning effect on some of these, there are interrelationships between selecting the temperament, actually selecting for it. And obviously when you, it's not a scientific program, seem to pick up so many beneficial effects. Mothering, well, we do those mothering ability, land awareness, all those things are part of that temperamental selection. I know I've never used the drug except years ago when we were trying to get letters to do it. But it's wonderful to have, and obviously the extrapolation to human systems is fascinating. But can you answer my first question at all? Can you see any linkage between temperamental selection and maybe its effects on other issues relating to stuff we talked about?
So broadly to reproduction, I mean, certainly we can see an effect of temperament, not within my own work, but it's more broadly recognised that that does improve reproductive potential both at the conception level and at the mothering level. In terms of, I suppose you're asking about a beneficial effect if we were to do that in combination with treatments like melatonin. I've never tested that. I suspect what would happen is because you're selecting to improve reproduction generally, that you would actually see the effects of drugs like that buffered because you have a higher standpoint or baseline to work off, if that makes sense.
It's a pretty silly question.
You just want an excuse to talk.
Sorry, Kelsey, just with the use of regular, did you do any dose response studies? Obviously, I'm assuming you use the commercial dose rate with the pellets that are in the dose, and obviously when you did double dose, so one after the other, seemed to mitigate the effects. Did you do any dose response studies?
We didn't do dose response studies. As you said, we were using the commercially registered dose for several reasons. One, using a dose response study means a lot more ethics and work on our end, which is a bit more time consuming. I think given what we've seen here, where we've got this sort of point where we can see an advantage, and then with that double dose, essentially a disadvantage or essentially a bring back, we probably wouldn't pursue dose studies given that we don't really want to go higher. We don't want to see that dichotomous effect of melatonin.
Probably thinking you want to go lower from a commercial.
Slightly lower from a cost point.
Return on investment.
Yeah. It's something to think about, but I think those effects that we're seeing are quite subtle. I mean, we're talking about a 6 to 8% difference in survival. It's not huge as it is. If we go lower, are we really going to try that? And it's something to think about. Maybe I'll break the implants in half or something. We'll see.
Yeah, I'm one of the non-academic friends, so sorry. You said there's a decline in, fertility in humans as well. Do you see that there's quite a strong link between, the dietary factors in sheep versus humans, or is it more cultural? Because I think there's probably a lot of cultural things that
Contribute to that.
as well.
Yeah, certainly. So, I mean, there's certainly cultures where those diets with phytoestrogens are much higher. I obviously don't specifically study humans, but I guess just within the literature, we do tend to see similar effects. I think what's more interesting about the human side of things is that actually isn't studied as in depth as what we can do in livestock. So there's a lot of studies where we've got almost an ambiguous result. They don't really know if it's driving it up or down. But in these model species where we can really get down to that molecular level, we have a lot of samples to work with. That's when we start to see much more clear effects. So I'd say the difference between these animals versus humans is really in the study design rather than the effect itself.
Kelsey, my studies have all been in the University of Hard Knocks, practicing farm. If I could ask a couple of elementary questions. Melatonin, is there any dietary correlation with melatonin? What that is, green feed versus dry feed, or seasonal behaviour? I know initially it comes from the sun. And then the other one is the agricultural chemicals that we are using in abundance. Have you found any correlations there with any of your work?
Okay, I'll try and answer your first one first.
I know the second question will be one.
Oh, that's okay, that's okay. So in terms of feed, I mean, we haven't specifically looked at this. We were just making sure that feed is balanced across all our groups, so we're not having an effect. I don't know about the effect of nutrition itself. I know or suspect that in undernourished ewes or use on a poorer feed basis, melatonin is more likely to have a pronounced effect. And again, it's exactly what we were talking about before with temperament or selecting for reproduction. If you've got a lower baseline to work off, you're going to see a more advanced increase in reproductive potential. As for your second question, so again, that's not something that I specifically study. Broadly, we know that a lot of these pesticides and herbicides do contribute to this infertility, and in fact, they act as endocrine disruptors as well. Slightly different mechanisms to what we see with these phytoestrogens, but they do also disrupt fertility in both livestock species and in humans as well.
Sure. Any other? Well, Kelsey, thank you very much for a terrific evening for all of us, and for the insights that you're making, and for the projects you're actually developing for the future, which I think gives so many alternatives for which to go. And at university, to provide the environment that allows this kind of work to flourish.
Can I ask one more, please?
You can.
Yeah.
I studied genetics at 1970 here, and all it taught me was that it's terribly complicated. It's evolving things. Now, epigenetic effects, I'm not even sure what they are. But when you were talking about your flies, and you got down to grandparent level, etc., was that just normal genetic way it worked, or was that epigenetic?
I suspect 100% it's epigenetic. I'm actually in the process of testing this. Given what we see in the sperm DNA with that fragmentation, what I've found so far in my epigenetic work in rams is that this is generally associated with an epigenetic malformation, essentially, with the sperm protamines and the histones. So I'm now starting to measure that in these flies in different regions of the reproductive tract to see if that's actually the case. But I think you're absolutely correct. In these cases where we're seeing this... Yeah, absolutely, of course. You sounded like you knew exactly what you were talking about. Yeah, I suspect that these epigenetic effects are really driving these effects broadly through these generations.
Thanks very much. Thanks, Kelsey.
All right. So